In this video, we will discuss about the life cycle of Plasmodium vivax. It is a malarial parasite, causes vivax malaria in human being. The life cycle of Plasmodium vivax is digeneric. It is completed in two hosts, primary host and secondary host. The organism which contains the sexual phase of the parasite is regarded as definitive host or primary host. In this case, as female Anopheles mosquito contains the sexual phase of the parasite, so it is regarded as the primary host or definitive host. And human, it contains the asexual phase of the parasite, so it is regarded as the secondary host or intermediate host. Life cycle of Plasmodium vivax is divided into asexual life cycle or zygogony in man and sexual life cycle or sporogony in mosquito. At first, we will discuss about the asexual life cycle or zygogony in man. Asexual cycle or zygogony in man is completed in following phases: infection, pre-erythrocytic zygogony, exoerythrocytic zygogony, erythrocytic zygogony, post-erythrocytic zygogony, and formation. Zygogony is the process of asexual reproduction by which Plasmodium vivax undergoes asexual multiplication. It occurs in human liver cells, which is known as liver zygogony, and in RBC, which is known as erythrocytic zygogony. Sporozoids represent the infective forms of the parasite. These are small, spindle-shaped, slightly curved or sickle-shaped, and uninucleate organisms. When a female Anopheles mosquito containing infective stages of parasite or sporozoids in its salivary glands bites a healthy person for sucking his blood, then the mosquito injects hundreds of sporozoids along with saliva into the bloodstream of the healthy person. The purpose of pouring saliva is to check clotting of blood as it contains an anticoagulant. When the sporozoids enter the blood, it remains active for about half an hour and disappears from the blood circulation. Then it enters into liver to invade hepatic cells. Here they multiply asexually by zygogony. Pre-erythrocytic zygogony and exoerythrocytic zygogony are two phases of liver zygogony. In pre-erythrocytic phase, each sporozoid grows for a number of days and becomes a spherical and non-pigmented zygote. It divides by zygogony and forms a large number of cryptomerozoids or cryptozoids. Their reported numbers varies from 1,000 to several thousands. They give pressure to the wall of the liver and liberate it out. It is the end of the pre-erythrocytic phase. This phase is completed within 8 to 10 days. In exoerythrocytic phase, the cryptozoids are ready to infect the fresh liver cell where they grow and become zygotes. The same process is repeated several times. The liberated merozoids in this phase are called metacryptozoids. The process of formation of metacryptozoids from the cryptozoids in liver cell is called exoerythrocytic zygogony. Two types of metacryptozoids are produced in this phase. Smaller and more numerous are micrometacryptomerozoids. They enter the red blood cells to start the erythrocytic stage. Larger and less numerous are macrometacryptomerozoids. They invade fresh liver cells to continue the exoerythrocytic zygogony. Both types of zygogony are continued side by side. Erythrocytic zygogony starts when the micrometacryptozoids enter into RBC. Single metacryptozoid enter into RBC and passes through trophozoid stage, signet ring stage, ameboid stage, and zygon stage. In trophozoid stage, inside RBC, the micrometacryptomerozoid becomes rounded with large nucleus and grows in size by ingesting hemoglobin of RBC. This stage of parasite is called trophozoid stage. The trophozoid grows inside a central vacuole is developed so that the nucleus is pushed to one side into peripheral cytoplasm. It resembles the signet ring with the peripherally located nucleus looking like the gem of the ring. This stage is clinically referred to as signet ring stage. Signet ring trophozoite ingests a large portion of cytoplasm of red blood corpuscles, forming a food vacuole into which it secretes digestive enzymes. The enzymes bring about proteolysis of blood hemoglobin, which breaks down into its protein component and hematin. Protein is used as food by the trophozoite, while the unused hematin forms the toxic malarial pigment called hemozoin. It induces high fever and shivering. The signet ring trophozoite develops into an active amyloid trophozoite. It sends seropotential processes into cytoplasm of the RBC. At this time, small red eosinophilic granules appear in the cytoplasm of the host corpuscles, which are known as Kapner's dots. The amyloid trophozoite, after active feeding, becomes rounded, grows in size, and becomes zygont. Asexual multiplication takes place in zygotes and forms merozoite. The merozoites are arranged to the periphery due to the presence of hemozoan at the center. The arrangement is just like the arrangement of petals of rose, so this stage is called as rosette stage. The merozoites give pressure to the wall of the weak RBC and liberate it out into the blood plasma in the form of erythrocytic merozoite. The erythrocytic zygote phase completed within 48 hours. Many merozoites enter the fresh RBC and repeat the erythrocytic cycle. Sometimes some merozoites produced in erythrocytic zygote reach the liver cell and undergo zygonic development in liver cells. This is referred to as post-erythrocytic zygote. Formation of gametocytes. After some generation of erythrocytic cycle, some of merozoites invade the new RBC. They grow in size but don't develop into zygon. Instead, they develop into gametocyte. The gametocytes are of two types: micro or male gametocyte and macro or female gametocyte. Micro or male gametocytes are 9 to 12 micrometer in length, motile, and few in number. They have large centrally placed nuclei. They take reserve food and stains faintly. Hence, the cytoplasm is light in color and clear. Macro or female gametocytes are large, 10 to 12 micrometer in length, numerous in number. They have small, compact peripheral nucleus. They have reserve food materials and the cytoplasm is dark in color. Further development in gametocyte stops in man, only possible in mosquito due to its low temperature. The life cycle of Plasmodium vivax is continued towards completion by following processes: ingestion, gametogony, fertilization, okinate, encystment, and sporogony. When a female Anopheles mosquito sucks blood of the infected person containing gametocytes in RBC, any other phases of parasite and RBCs are digested. The gametocytes, however, survive. These are liberated and become lodged in the cavity of gut. Development of gametes from gametocyte is known as gametogony or gametogenesis. Gametocytes, the gametes are also of two types, microgametes and macrogametes. The male or microgametocyte begins to undergo a process called exflagellation. In each microgametocyte, nucleus divides by mitosis to produce 6 to 8 haploid daughter nuclei, which assemble at the periphery. The cytoplasm outgrows into long, thin, and flagella-like projections, and a daughter nucleus enters each one of them. These projections break away as mature male or microgametes or sperms. Female megagametocyte undergoes some reorganization and becomes a female gamete, that is megagamete or macrogamete or ovum, which is ready for fertilization. One microgamete penetrates into macrogametes through the cone of reception, and fertilization takes place known as syngamy. A complex fusion of nuclei and cytoplasm of the two gametes occurs, resulting in the formation of zygote with a single diploid nucleus or synkaryon. After fertilization, zygote remains rounded and non-motile for some time. Then it becomes elongated and vermiform, known as ukinate. Ukinate is motile and has pointed ends. It penetrates the wall of stomach with the help of lytic secretion. It settles down just under the thin membrane that separates midgut from the humuscle. The cyst wall is thin, membranous and elastic, secreted partly by ukinate and partly derived from midgut tissue of mosquito. The encysted zygote is called oocyst or sporont. Now enters a phase of asexual multiplication known as sporogony. It is the process of formation of sporozoid from zygote. Oocysts mature and develop. The nucleus of oocyst divides first by meiosis and then by mitosis, forming large number of haploid nuclei, and forms sporozoid forming cell known as sporoplast. The nuclei of sporoplast again multiply and cytoplasm get constricted around them. Thus the result and structure in the sporoplast elongated to form a slender or sickle-shaped sporozoid. In this way, 10,000 sporozoids are produced from each oocyst. Therefore, each oocyst fills with numerous sporozoids. Now this gives pressure to the oocyst, and due to this, the oocyst ruptured and thousands of sporozoids are released in the body cavity humuscle. The sporozoids are very active, motile, so reach to the salivary gland of the sporozoids are ready to infect the healthy person after each bite of mosquito. When a mosquito bites a healthy person, thousands of sporozoids are injected into the blood along with saliva.